Hello everyone, today our group will be presenting about nucleic acid technologies. So the first one is PCR or polymerase chain reactions. So PCR is a revolutionary technique in molecular biology, often referred to as a photocopier for DNA because it allows us to amplify a specific DNA fragment into millions of identical copies within just a few hours. The principle of PCR is based on a natural DNA replication, but carried out in vitro. The process relies on repeated cycles of heating and cooling, which enable three key steps: denaturations, in which the DNA strands are separated when they break the hydrogen bonds at 95 degrees Celsius, annealing, where the primers adhere to DNA strands at 55 degrees Celsius, and the extensions, where the Taq polymerase builds complementary strands at 72 degrees Celsius. And then to carry out PCR, we use thermal cycler, also known as the PCR machine. It holds the sample in small tubes and automatically changes the temperature according to the program cycle. This precise control is what makes PCR fast, accurate, and reproducible. The uses of PCR in forensics is to comparing unknown sample to known samples or a database. In agriculture, it is used to plant genotyping and plant cloning, and in medicine, it is used for genetic testing, treating cancer, and COVID testing.
So what techniques to separate DNA fragments? So we can use gel electrophoresis, where the principle is the separation of charged molecules based on their size and charge using an electric field within a gel matrix. So the apparatus of these tools consists of buffer solution, sample slots, bands, gel, and also the electrode. We can use the agarose that looks like a web, and the web acts like a size filter. There is a pocket called wells allow us to add our samples into the gel. And there is a metal wire that placed on either side of the gel to act as the positive electrode and the negative electrodes, so the electricity can flow between them. And gels are submerged in a conductive liquid buffer to conduct the electricity between the negative and positive electrodes. And what happens when we run samples on a gel? So the molecules are attracted to the opposite charge. So the negative molecules move towards the positive electrode, while the positive molecules move towards the negative electrode. And the smaller molecules move more easily to the web and travel faster, while the larger molecules have more trouble moving through the web and travel slower. So the samples are placed in the well at the start of an experiment, and we can use color diodes to differentiate it, and the power is turned on and samples travel into the gel. As they move through the gel, the molecules travel in a straight line, and it looks like a small horizontal line that we call a band. What is the charge of the molecules that were in the sample? So the green molecules in the sample move towards the negative electrode, it means it is positively charged, while these blue molecules towards the positive electrode, it means it is negatively charged. And what is the composition of the molecules that were in the sample? So this lane has a single green band, it means the sample contains a single type, while this lane has two bands, it means the sample contains two types of molecules. And how do scientists use gel electrophoresis? So the gel electrophoresis is most commonly used to separate and visualize DNA samples, and also used to identify people based on DNA, diagnose genetic diseases, and more. And the result of the DNA gel electrophoresis is look like this in the real life.
Alright, I'm Raka Putra Davianda. I would like to continue about Sanger sequencing method. The Sanger sequencing method is a classical technique to determine the order of DNA bases, ATGC. So the principle of the Sanger sequencing is the first one is chain termination, while the NTPs or dideoxynucleotides hold DNA synthesis at specific bases. The second one is DNA polymerase, which is DNA polymerase extends the DNA strand until the NTP is incorporated. And the last one is fragment separation, which is fragments separated by size using gel electrophoresis. In chain termination, as we know that 3-hydroxyl group in nucleic acid is playing role to DNA polymerization, forming phosphodiester bond. But if 3-hydroxyl group is absent, the DNA synthesis will stop. This molecule is called the dideoxynucleotides or DDNTPs. The second one is DNA polymerase. The DNA polymerase can extend the primer DNA using the DNTPs and occasionally stop the DNA synthesis with the DNTPs. And the last one, fragment separation. The fragment will be separated by polyacrylamide gel electrophoresis. So the single sequencing result, the DNA sequencing result will be represented by chromatogram. By each nucleic acid is represented by different color. The application of DNA sequencing in medicine and health to detect genetic disease, in forensic individual identification, and for biology and biotechnology is in gene function research. So the next generation of sequencing methods, the first one is Illumina sequencing. It determines the DNA sequence by adding nucleotides but one by one. And for ion torrent sequencing, it determines DNA sequence by measuring pH changes. When nucleotides are incorporated, it doesn't use a fluorescence label but relies on semiconductor sensors.